Thank you very much. I'd like to introduce myself. I'm Allison Hubble. I'm the Chief Scientific Officer, as indicated before. And I'm going to talk to you today about how we're developing better and safer therapeutics by improving the cryopreservation of cells. And we're going to start at the end. We're going to start talking about the patient experience. And in this slide, you see a juvenile recipient receiving an infusion. How many people in the audience have been present when a patient has received either a hematopoietic stem cell product or a cell therapy? Okay, then you may have a cryopreserved one, and then you've seen the adverse reactions and smelled the metabolism of DMSO on the floor. So we're going to work towards eliminating that. Everyone at this conference is interested in cell therapies becoming the fourth pillar of medical care to add to pharmaceuticals, biologicals, and medical devices. In order to achieve that outcome, we need four things. We need scalable manufacturing. We need the ability to have those products approved. We need to have insurance companies or a payer pay for them. And then finally, we need physicians to adopt them for a standard of care. And preservation is a critical element in the manufacturing infrastructure. Why is that the case? The supply chain for cell therapies is unlike any other supply chain for any of those other pillars that we talked about in the previous slide. Minnesota, where we're from, is the home of over 400 medical device companies, and I can tell you the supply chain for a medical device company is completely different than cell therapies. Specifically, we have cells collected typically from a healthy donor or a patient in one location. It's then sent to the site of manufacturing. It could be genetically modified, it could be expanded or selected, and then it's transported typically to a third location where it may be administered. All along this supply chain, the cells need to remain viable and functional. So what we know is that existing cryopreservation solutions have three basic pain points. One, they make the manufacturing process more difficult because exposure to DMSO results in cell losses. DMSO may not work for every cell type, and potency is at risk. And thirdly, we have the infusion-related, especially in certain organ locations and indications such as pediatric patients. So we dug into this more deeply before we started the company, and we did customer discovery using funding from the National Science Foundation. And we interviewed over 200 stakeholders in the cell therapy area. We interviewed everyone from physicians who administered cell therapies to heads of manufacturing of developers. And what we saw agreed with that previous slide. There were concerns about cell performance, in other words, the number of cells that survive and their function. There were certain groups that were very concerned about the manufacturing issues associated with the use of existing methods. And the third, patient safety. So those three pain points. So I was in my office in Minnesota in a lovely February, and it was freezing cold, and I was looking out the window, and I said, how do trees survive Minnesota winters? And the answer to that question, big surprise, is they express molecules to help protect them against the cold. These molecules are known as a category as osmolytes. So I had a hypothesis that combinations of osmolytes, nature would never just use a single molecule. Nature would always use combinations of molecules to stabilize a biological system. So we hypothesized that sugars, sugar alcohols, and amino acids will be effective in preserving cells. One of the tools I developed in my lab was low-temperature Raman spectroscopy. I'm trying not to get too technical here, but this is a really cool tool because we can cool cells at a controlled cooling rate to a controlled temperature and interrogate them using spectroscopy, using a laser, at low temperatures. Now, we don't want to get into a philosophical conversation about whether a frozen cell is alive or not, but we can determine a host of signals that are critical to understanding the cell response. Where is there ice? Is there ice inside the cell or only outside the cell? Where is the cell in this whole field of view? Where are the proteins? Where are the cryoprotective agents? So we can determine, based on chemical signal, the location of cryoprotective agents, the locations of proteins, the location of the nucleus of the cell. So it's an extremely powerful tool. It's extremely data rich, and we're even interrogating data sets we've gotten a decade ago to see more signals. So how can we use that to prove our hypothesis? Well, we did imaging of sugars and the manner by which they interact with the cell. In this image, we show images rendered during freezing of a cell, and we mapped the sucrose in the solution. And what we found is there was overlap between the sucrose and the cell membrane. The sucrose doesn't enter the cell, but it intercalculates in the cell membrane. And what we did with these studies is we actually established experimentally proof that sugars interact with the cell membrane to help stabilize it. This theory had been put forth in the 90s, and we were the first to establish experimentally that this hypothesis was true. So we know one of the methods of action for one of our components is to stabilize the cell membrane, a critical structure for the cell. But look, there's more. Sugars and sugar alcohols interact with water, and they form what is called a naturally occurring deep eutectic system. And what that means is the hydrogen bonding between these three molecules, the sugar alcohol, and the water, is so tight that the volume that that complex takes up is less than the volume of the individual molecules. Well, big whoop-dee, who cares about something like that? What it does is it influences, that molecular level interaction influences the macroscopic freezing behavior of that solution. So by freezing a naturally occurring deep eutectic system to a given sub-zero temperature, we have less ice present. That means we have protected the cell from the adverse effects of that ice. It also changes other entities such as viscosity, but those have not been problematic when processing cells. Okay, so we have three components. We could spend decades and thousands of experiments optimizing. None of us in the room have the ability to take that time and money to do that. So we have an AI-driven algorithm that allows us to optimize in this parameter space with a small number of experiments. The cells have much to teach us, and we have to take that information, whether a cell is viable or non-viable, and translate that into an outcome that improves viability. And we do that through the algorithm in as little as 4 experiments and as much as 7 or 8. So we've reduced the number of experiments to optimize these compositions from tens of thousands to less than 10. So let's do a side-by-side comparison. What we're talking about is natural components that do not involve risk to the patient. We can get high post-thaw recoveries. It's not important just to get high post-thaw recoveries, but it is important to have a consistent outcome. You can't, if you're using this as a drug product, you need to have high viability and a consistent viability. Finally, we improve the manufacturing process. We do not require washing. We do not involve any leaching of plastics due to solubility. And we give you an increased time for stability of the cells in solution prior to freezing and post-thaw so that you can build this process into your manufacturing setting and the clinical setting. So I'm going to talk in the next few slides about some head-to-head comparisons we've done, because the proof is never what happens in my academic lab, but what happens out in the field. So the first cell type we worked on was mesenchymal stromal cells. It's important to understand when we look at the efficiency of freezing and thawing, we don't look solely at viability. We have to look at recovery. Some people may call it yield. But what that does is it helps you capture the effect of freezing and thawing on your cells, because a certain population of your cells will actually lyse as a result of the freezing process. So recovery eliminates the ability to bias that measurement. Now, MSCs are not effective unless they are actually attached to a surface so that they can provide the immunomodulatory or whatever their clinical function is. And what we have observed in DMSO-containing solutions is DMSO actually damages the actin cytoskeleton of the cell and interferes with its ability to attach. But when we take the sugar alcohols, and amino acids, we get high degree of attachment and post-thaw function. And we've actually done RNA-seq on these cells, and they express cytoprotective genes and not stress-related genes. But look, there's more. We've also done some work with some developers who were looking at a CAR T product. And you'll note that we looked once again at donor-to-donor variability and also recovery. And we compared head-to-head with CS10, and we had higher rates of recovery for a CAR T product. We're also looking at an NK cell product. With NK cells, they've been very difficult to cryopreserve. We've had very high levels of post-thaw viability recovery, post-thaw, and up to 24 hours post-thaw, so they persist as well. So this is kind of a representation of the algorithm and the way it operates. This is for optimizing the cryopreservation of induced pluripotent stem cells, a very important cell source for different cell therapy and regenerative medicine applications. As we go through the algorithm and optimize, what you'll notice is the solution compositions in green have post-thaw attachment equal to or greater than 7% DMSO. And so we are able to produce a family of solutions that have high performing characteristics. And we looked at something like attachment in order to have a more functional metric for the iPS cells. If they don't attach post-thaw, they can't go on and differentiate. And another unique attribute is some of our cell therapies and some of our source materials represents heterogeneous populations of cells. We can use this algorithm to actually optimize the preservation of those heterogeneous cell types. And this is just one case study. So what we noticed when we looked at PBMCs is we're actually seeing a diminution of the CD8 positive fraction compared to the CD4 positive fraction. And we said, let's use our algorithm to try and achieve an outcome that will enable us to have a high level of CD4 recovery and a high level of CD8 recovery. And we went through a series of a whopping 4 experiments, and we were able to improve the overall recovery of the CD8 fraction so that we're not seeing a depletion of one cell type versus another. Because once again, it's not just that high recovery. You are relying on a drug product in which those two components each have a therapeutic benefit. If you're depleting one population versus another by the freezing process, you are impeding the effectiveness of your drug product. So we can use an algorithm to help optimize that as well. So how do you work with us? We have worked on a variety of different cell types that are given in the purple box here, and we're continuing to develop more experience with other cell types as time goes on. We are interested in partnering to look at feasibility, to look at optimization, and to look at supporting you and your preservation needs through your IND-enabling support. And most importantly, because we all know we're getting tired after God knows how many presentations and personal meetings, we have a social event going on right now in the bar to announce the formation of CryoX, or a Center for Cryo Excellence in Houston. And we want everybody in this room now that, am I not the last speaker in this session? Not yet? Oh, we want you to, oh, okay, so stay for the next speaker. And join us in the bar to celebrate the opening of this. Thank you very much, and I'm happy to take whatever questions you have in the last 20 seconds. Okay.